Okay, so in chapter 4, we'll talk about cell injury, aging, and death. Now, the first forms of cell injury we'll talk about are the reversible forms of cell injury. And one of the first signs of cell injury that's reversible is something called hydropic swelling. Really, hydropic swelling is an accumulation of water inside of cells due to the breakdown of the sodium potassium pump. So in previous chapters, we've talked about how the sodium potassium pump is an ATP-driven pump that helps pump 3 sodiums out and 2 potassiums in. Now, by pumping ions out of the cell, you're basically helping to establish a certain concentration gradient. Now, if your cells are stressed and have some injury, and they're not able to make as much ATP, and the sodium potassium pumps start to break down, well, then you're not pumping as much sodium out of the cell, and so water via osmosis will start to be attracted into the cell where there's a higher concentration of solutes like sodium. So that as a consequence of lack of ATP, the sodium potassium pumps start to stop pump sodium out, and that helps bring water into the cell, which causes the entire cell to swell. This is called hydropic swelling. And what's interesting is that hydropic swelling amongst all of the cells in an organ or tissue can actually lead to visible changes in the size of that tissue. If the liver is injured, you can start to see the liver start to actually swell up because all of the cells in the liver start to accumulate water via this hydropic swelling mechanism. Now, as long as those cells don't swell to the point where they burst, this is a reversible form of cell injury because all you really need to do is reestablish enough ATP to get this sodium potassium pump working again, and that can help reverse the swelling that we see. Now, what the slide shows is basically hydropic swelling, where this is actually the kidney tubules. So if you look at these simple cuboidal cells of the kidney tubules, they have kind of a nice cube-like appearance. But if you look over here, what we're looking at is basically hydropic swelling once those cells become sort of injured. You can see that the cells become much larger here. They have more pale and diffuse cytoplasms. In fact, it looks like some of these cells may have already burst. And if they burst, that's actually an irreversible form of injury. But as long as they just swell and don't burst or lyse, then that should be reversible. Like this cell still appears to be intact. So as long as ATP gets reestablished in that cell, then you can reverse the swelling and help sort of maintain that tissue. Now, this is going to be an accumulation of water. So you have these large, pale cytoplasms with also altered organelle appearance too. So the endoplasmic reticulum is going to be swollen, as well as the mitochondria will be swollen, because again, these organelles also have sort of compartments within them that can swell with water. So they will also swell with water when a cell becomes injured. Now, this can lead to the increase in the size and weight of an entire organ, which we call megaly. So, for instance, if the liver becomes sort of injured, that can lead to hepatomegaly, which is basically a swelling of the liver. Or if the heart becomes injured, that can lead to cardiomegaly, which is basically a swelling of the heart. And it's a form of reversible injury. Now, another sign of cell injury are the accumulation of intracellular debris. So a lot of times when cells are stressed, they can start to accumulate substances within the cells, and that can occur during injury from toxicity, immune response, or just debris that might be taking up some cellular space. Now, these intracellular accumulations are really characterized by excessive amounts of normal intracellular substances. So some examples of things that might accumulate in excess could be things like fatty acids in liver cells, maybe misfolded proteins in different types of cells, or accumulations of exogenous material like metals, that kind of thing. Now, what these are showing are just some different examples of accumulations that can occur. So with this example, what it's saying is that if you have altered metabolism in liver cells, that can actually cause what's called fatty liver, where liver cells can start to uptake a lot of basically fatty acids, and they can start to appear like adipose, or they can start to kind of look like adipose cells. So it can cause the liver to actually go from a kind of a nice gray appearance to kind of an odd yellowish white appearance. and that's when the liver becomes reversibly damaged, or even irreversibly damaged. Now, what can also happen too is if a cell is stressed, it might not fold proteins in the right way, so you can get these aggregations of misfolded protein that can accumulate inside the cell. That can place further stress on that cell and lead to further damage. Or what can happen too is if a cell is stressed, then you might actually not create enough enzyme that breaks down a certain product, and that itself can lead to an accumulation of debris in the cells due to the lack of appropriate enzyme to remove that debris. Another example too is that when cells are stressed, they can start to uptake extracellular material, like especially things like metals. And so in stress cells, you might see an accumulation of something like iron or copper. And we'll talk about some different hereditary diseases where certain cells can actually uptake a lot of these extracellular materials. Now, what this is showing is basically in lung tissue how when lung cells are stressed, they can start to uptake a lot of extracellular debris, even like carbonaceous material from cigarette smoke can start to be taken into the cells of the lung tissue. And you can see how the lung tissue is actually kind of stained darkly up here superiorly, and especially by the hilus of the lung. In this person, I mean, this is obviously a deceased individual because we're looking at the cross-section of the lung, but you can see there's all this debris that's being taken into the tissue, and it's actually caused scarring of that tissue, which we call anthracosis. Down here is sort of a more normal, healthy-looking lung tissue. But you can see it looks dramatically different from the tissue up here, which is more stressed and scarred from the uptake of extracellular material. Now, another example of sort of adaptation to cell stress is something called atrophy. Now, atrophy occurs when cells shrink and reduce their sort of size and function due to injurious factors. And there's a lot of different things that can cause atrophy of cells. But some general causes of atrophy can lead to things like, or can be caused by things like disuse or lack of use of a tissue can lead to atrophy of those cells. Lack of nerve supply to the tissue can lead to a decrease in the activity and size of the cells, as well as things like ischemia, which is basically lack of blood flow to a tissue, nutrient starvation, interruption of endocrine signals or hormone signals, persistent cell injury, or even aging. But all of these are actually causes of atrophy, which can lead to a decrease in the size of cells and even a decrease in the entire organ. So, for instance, you'll be familiar with individuals who are bedridden. They can get a lot of disuse atrophy of their skeletal tissues because if they're just laying in bed all the time, they're not using those skeletal tissues and they can become weaker and really make that individual more prone to further injury. Now, what this slide is showing are some different forms of cellular adaptation. Yes, we talked about atrophy, which is a decrease in the size of normal cells. You can see that the normal cells here are larger than the atrophied cells because atrophy means shrinking of cells. But what can also occur during cellular stress are things like hypertrophy, which is an increase in the size of the cells, hyperplasia, which is an increase in the number of cells, but that can also lead to things like metaplasia or dysplasia. Now, hyperplasia can come about from sort of a chronic stress on a particular tissue where the cells start to increase in number. What can also happen too is that with chronically stressed tissues, the cells can start to change their type in order to better suit the stresses placed in that tissue. So an example of hyperplasia that can occur in the body would be like with gastroesophageal reflux disease, when there's acidic fumes going up into your esophagus. That can burn the lining of your esophagus, causing those cells to increase in the number. By having a thicker epithelium, it can better resist those stomach acids. Now, in the long term, that can lead to things like metaplasia, where the epithelium might change from one type to another, and that can lead to a type of precancerous growth called dysplasia. And dysplasia is basically disordered growth of cells in a tissue, which is often precancerous. So what's odd about stress on a tissue is that chronic stress can lead to dysplasia, which is precancerous growth. So some examples of this is, for instance, smoking, which is the chronic irritation of the airways. That can put you at risk for things like airway cancer, because that's chronic irritation that can cause dysplasia. Or people who swim a lot, especially in pools with chlorine, they're more prone to skin cancer because that chlorine is irritating to the skin. Now, hypertrophy is just an increase in the cell mass, but without an increase in the number of cells. So, for instance, sometimes if there's a lot of stress placed on a particular organ or tissue, those cells can increase in size without increasing in number to better try to accommodate for that stress. An example of hypertrophy that we'll talk about later on in the class is cardiac hypertrophy, where if the heart has to work harder to pump blood throughout the body, it'll increase in size by increasing the size of cells. Cardiac cells will increase in size. And in fact, that's what you can see on this slide here, where this is kind of more of a normal heart. This is a hypertrophied heart. You can see that the wall of the left ventricle is much thicker in the hypertrophied heart because the cardiac cells have to work much harder to pump blood out of the heart. And so this is really a sign of decreased cardiovascular health, where if the heart has to pump more strongly to get blood circulating, then those cells can increase in size, which can lead to hypertrophy. You can see that there's really not much left for the ventricular space, and so that makes it even more difficult for the heart to pump blood. Now, hyperplasia is an increase in the number of cells in your tissue, and this is basically usually going to come about from increased physiological demands or hormonal stimulation. Like if a organ or tissue is being stimulated more so by a hormone or has to be used more often, that can induce hyperplasia. Metaplasia is going to be due to persistent injury or chronic injury, how one tissue type can actually change into another. So meta means new, metaplasia means changing a tissue type to another. So an example of metaplasia would be how the pseudostratified ciliated columnar epithelium you find lining the airways in most people, in smokers, can be changed to a form of stratified squamous epithelium, which lacks those cilia. Now, the stratified squamous epithelium actually comes about from metaplasia, and although it can help protect that individual's airways, it doesn't help remove the debris from those airways. And so that's a form of metaplasia, but that can actually lead to sequelae that lead to further tissue damage, because now those individuals will start to retain those basically carbonaceous material from their cigarette smoke. Dysplasia is actually a disorganized growth that's often precancerous. So typically what happens with chronic stress on a tissue is you can get metaplasia, that can lead to dysplasia, and that dysplasia can lead to cancer. So chronic irritation of a tissue, which leads to dysplasia, can be precancerous. So an example of this would be, let's say if you're out in the sun a lot and you get metaplastic growth of tissue, like which causes a mole to form. If that mole leads to dysplasia and that dysplastic growth breaks away and spreads to other parts of the body, well, that could be cancerous. So it's kind of odd how something like skin irritation from sun can lead to dysplasia or disorganized growth that can lead to cancer. Now, necrosis is actually a form of irreversible cell injury, and this is going to usually come about as a consequence of something like ischemia, which is lack of blood flow, or toxic injury, due to exposure to chemicals or heat or radiation. And necrosis is really characterized by cellular rupture, which leads to spilling of contents into extracellular fluid, and that causes an inflammatory immune response. But there's different mechanisms of necrosis that we'll talk about. Now, what's common though with all these forms of necrosis is at the cellular level, what can happen is that we can start to see these blebs that form during reversible cell injury. And these blebs are more consistent with hydropic swelling. But if that form of cell injury becomes more severe, what happens is that the plasma membrane starts to break down. Organelles start to break down because they swell too much. The nucleus starts to explode as well because it swells with too much water. And really we reach a point which is almost like allostatic overload on cells, where we can't really return to homeostasis because too many of our mechanisms are overloaded and they can't reverse these injuries So we get this irreversible injury, which is going to lead to necrosis. Now, there's 4 main types of tissue necrosis we'll talk about. The first of which is coagulative. Now, coagulative necrosis begins with ischemia, or lack of blood flow, and that ends with degradation of the plasma membrane. So coagulative necrosis is caused by ischemic events, that could be blood vessel rupture, or blockage of a blood vessel, or decrease in blood flow, that basically leads to degradation of that tissue in the long term. Now, liquefactive necrosis comes about when dead cells actually dissolve and they become liquefied by enzymes in those cells called lysosomal enzymes. Now, typically, liquefactive necrosis causes abscesses or cysts in this dissolved or dead tissue. And so, an example of liquefactive necrosis is typically with brain damage. The brain tissue will degrade in a liquefactive necrosis manner. So typically, the brain will form these large cysts that are full of basically the breakdown products of dead cells. However, liquefactive necrosis can occur in other parts of the body. For instance, influenza, in some of its more early forms back in the early 1900s, when we had the influenza outbreak that killed millions of people worldwide, that form of influenza virus was so virulent, it actually caused liquefactive necrosis in people's internal organs. And when they did autopsy on those individuals, basically their internal organs were just mush. They were just liquefied from this virus because it caused a form of liquefactive necrosis. Now, the form of influenza virus that we live with now today is less virulent, and it doesn't cause as much liquefactive necrosis. You don't see that liquefying of organs that you saw 100 years ago. But who knows? Maybe the virus could revert back to its old form one day. Now, fat necrosis occurs when we have death of adipose tissue. It's usually the result of trauma to adipose tissue or pancreatitis, which is basically inflammation of the pancreas. What happens is that with this fat necrosis, these enzymes get released that start to break down the fatty acids, but also those fatty acids become mineralized by some of the ions you find in cells. And what's interesting is that fat necrosis forms these chalky white areas of tissue which resemble soap. In fact, fat necrosis actually causes a form of saponification, like the way that soap is formed, because really soap is just mineralized fat. And so fat necrosis is like a saponification of your body's own adipose. And I'll show you guys a picture of this coming up. Now, caseous necrosis is a form of necrosis that resembles clumpy cheese, because caseous means cheese-like. And so caseous necrosis kind of resembles clumpy cheese. An example of this is actually the form of tuberculosis infection that forms these caseous necrosis cysts in your lungs. And so this example here is showing coagulative necrosis, which is a form of gangrene in the foot here. This would be an example of dry gangrene, where due to lack of blood flow, the tissue starts to break down, the plasma membrane starts to break down, and it forms this coagulative necrosis of the distal extremity here. Now, this is showing a form of liquefactive necrosis, where the cells start to break down and they form these large cysts. And inside of these cysts, you'd find the breakdown products of those dead cells, where lysosomal enzymes basically break down the cells that are dead. And so what you end up with then is this sort of soup of material from the dead cells that forms a cyst somewhere in the body. Now, if you all remember, fat necrosis form these soap-like deposits in the tissue. So each of these little white specks is basically going to be the fat necrosis we see here, where enzymes that break down adipose basically break down those fatty acids into kind of more simple pieces, which get mineralized by your body's own ions, and it forms soap-like deposits in the tissue, which we see here. That's fat necrosis. Now, caseous necrosis is common in the lungs, but can occur elsewhere too. But it forms these cheese-like deposits, kind of like a thick, clumpy cheese in tissues. And this is an example of caseous necrosis of the lung here. Now, gangrene is also a form of irreversible cell injury. And gangrene can basically occur when we have cellular death in large areas of tissue. Now, it results from interruption of blood supply to tissue. So we're talking about a form of coagulative necrosis or ischemia. But there's different types of gangrene. We have dry gangrene, which is basically a form of coagulative necrosis. That we have blackened, dry, wrinkled tissue that has a demarcation from healthy tissue. That's dry gangrene. Now, wet gangrene is actually a form of liquefactive necrosis that's typically found in internal organs. So, for instance, this wet gangrene would be what would be caused by influenza virus, like I mentioned earlier, where we have liquefactive necrosis in internal organs. Gas gangrene is actually an accumulation of gas within the tissue that's typically caused by necrosis or death of tissue that coincides with an infection, especially an infection of like Clostridium bacteria. Like if you think of like Clostridium botulinum or Clostridium tetani, those are forms of bacteria that sort of in excess in infection can lead to gas gangrene, which is basically an accumulation of gas in that tissue. Now, there's another form of irreversible cell injury, which is actually due to a form of programmed cell death. This is called apoptosis. Now, apoptosis is actually a form of programmed cell death that occurs not necessarily as a result of cell injury, but can be an important component of just normal development. And I'll give you guys an example of apoptosis. When your digits form, like your fingers and toes, all of your digits are actually connected by a thin piece of skin between each one. And so if you think about that, it kind of looks like a webbing. Now, normally that webbing goes away prior to birth because those cells between the digits, they undergo apoptosis, which is a form of programmed cell death. That way, you're born without that webbing. However, if that apoptosis fails to properly occur in those cells, then people could be born with things like webbed feet or webbed toes. So it's not always a pathological process. It does not cause inflammation. But apoptosis can occur due to cell injury, where if a cell is chronically stressed, the cell can actually just kind of kill itself through a process called apoptosis. So back when we talked about coagulative necrosis, we talked about how that's typically caused by ischemia. And I mentioned how ischemia is due to a lack of blood flow. Now, hypoxia basically just is a word to describe how there's low oxygen levels in a tissue. But typically, tissue hypoxia is caused by ischemia. So usually, if there's low oxygen levels in a tissue like blood, this is due to a decrease in blood flow, which is ischemia. However, there are certain forms of injury where hypoxia can be caused by something else. Not a lack of blood flow, but maybe inhibition of normal oxygen transporting mechanisms. Like for instance, carbon monoxide can cause hypoxia, but not affect blood flow because the blood can just still continually flow how it normally does. But carbon monoxide can block hemoglobin's ability to transport oxygen, which can cause hypoxia or low oxygen levels in the tissue. But typically, tissue hypoxia is caused by ischemia. Now, ischemia is the most common cause of cell injury, and the reason why ischemia is more damaging than just hypoxia is that a decrease in blood flow not only doesn't deliver oxygen, but also doesn't remove wastes from the tissue and also doesn't deliver other nutrients like glucose or fatty acids or protein. So ischemia is much more damaging than hypoxia alone. Now, and this is because, like I said, it's a combination of disrupted oxygen supply and an accumulation of metabolic wastes like lactic acid, and that could damage the tissue further. So even if someone has low oxygen levels in the blood, as long as their blood's still moving, that's less damaging than low oxygen levels in the blood and stagnant blood, or basically blood that's not moving through the tissue. Now, what happens is that if lactic acid does build up in the tissue, that can lead to lactic acidosis. Now, because lactic acid's an acid, that can lead to further damage of proteins and other molecules in that tissue, so that these enzymes can become more dysfunctional, and that can lead to further tissue damage. Now, up to a point, ischemic injury is reversible, but cell death is going to occur like if the plasma membrane breaks down or other organelles become critically damaged. Typically, most tissues in your body can survive about 4 hours under ischemic condition. So, for instance, when they do like carpal tunnel surgery to help out with carpal tunnel, they can cut off blood flow to the hand for up to 4 hours, and the tissues of your hand are going to be just fine up until that point because they can survive 4 hours without blood flow. However, about after 4 hours or so, that tissue starts to break down. Other tissues, though, like your brain, are less able to cope with ischemic events. So the brain tissue doesn't last 4 hours with the lack of blood flow. It's going to last much shorter period of time. But what happens with ischemia is that this low oxygen levels leads to an increase in the amount of calcium you find inside the cell. Okay? Once calcium starts to accumulate inside the cell because of a decrease in calcium pumps, this leads to the activation of different enzymes like proteases and phospholipases. Proteases are basically enzymes that break down protein like the cytoskeleton, and phospholipases are basically enzymes that break down phospholipids like your plasma membrane. So due to an accumulation of calcium inside the cells, that leads to the breakdown of cytoskeleton and plasma membrane, which, if severe enough, could lead to irreversible cell injury and death. What can also happen too is that when the cells become ischemic and there's this damage occurring, that can recruit immune cells into that local area of tissue damage, and these immune cells start to release what are called free radicals, which can start to also break down that cell, because the injured cells can recruit your own immune system to start to break them down, and that can lead to further membrane damage and possibly even irreversible cell injury and death. Now, there's other forms of cellular injury like nutritional injury. Nutritional injury comes about when there's not enough nutrients to support normal cell growth and development and just metabolism. So, not having an adequate amount of fats, carbohydrates, and proteins or vitamins and minerals, which can lead to decreases in normal cellular function. Typically, nutritional injury results from poor intake, altered absorption, impaired distribution by the circulatory system, or inefficient cellular uptake. So, for instance, an example of inefficient cellular uptake of a nutrient could be like with type 2 diabetes, which is insulin resistance. Because the cells stop responding to insulin, many of the cells of your body stop being able to transport glucose into the cell, which means that they're not efficiently uptaking that nutrient, and so that can lead to further nutritional injury of that cell in your body. Now, some common causes of malnutrition could be things like poverty, alcoholism, acute or chronic illness, self-imposed dietary restrictions, and malabsorption syndromes. Now, poverty is associated with malnutrition because typically if someone has poverty, they probably aren't eating properly. They're not getting like a well-balanced diet. They might be living off of just like rice and beans and not getting all of the nutrients they might need. Now, chronic alcoholism can lead to malnutrition because alcohol can irritate the gut, making it more difficult to absorb nutrients. But other acute or chronic illnesses, which may decrease your appetite or make it more difficult to absorb nutrients, can lead to malnutrition. Otherwise, self-imposed dietary restrictions are a common form of malnutrition as well. And other malabsorption syndromes like Crohn's disease, which is an inflammatory disease of the gut, can lead to malabsorption. Now, there's also infectious and immunological injury, which can come about. And this can be caused by, sorry, I'm laughing at my dog. This can be caused by bacteria and viruses your cells in a variety of different ways. Get out of here. Come on. Now, depending on its virulence. So, like, bacteria or viruses may injure a cell depending on its own virulence. So, some bacteria are actually intracellular parasites, and some viruses can actually cause injury or damage to the cells. Now, chemical injury can be caused by toxic chemicals or poisons causing cellular injury. An example of chemical injury could be something like arsenic exposure. It turns out that arsenic inhibits the electron transport chain, and that can actually lead to cell injury and death. Otherwise, there's other forms of physical mechanical injury, like extremes in temperature, abrupt changes in atmospheric pressure, like if you're scuba diving, you get the bends. Otherwise, things like mechanical deformation, electricity, and ionizing radiation are all forms of physical and mechanical injury. Now, we'll focus on this ionizing radiation part next. Now, what ionizing radiation refers to is radiation exposure. And really, there's lots of different types of radiation. And not all radiation is necessarily bad for you. What this is showing is the whole electromagnetic spectrum that we know about. And this ranges from things like audio frequencies to radio waves to microwaves, all the way up to things like infrared, the visible light spectrum, and then including things like ultraviolet, X-rays, gamma rays, and other cosmic rays. Now what happens is that what we see here is a scale of wavelength. So that the longest wavelengths are the audio frequencies. Those are going to be part of the electromagnetic spectrum whose wavelengths can be up to like 10 billion meters long. Otherwise, that can vary to wavelengths that are microscopic. When you start talking about like cosmic rays, those have very, very small wavelengths. Now, it turns out that the smaller and smaller the wavelength, the more potentially damaging that radiation could be to your body. So we see that audio frequencies, radio waves, microwaves, infrared, and visible light, they're going to have fairly short wavelengths. Fairly long wavelengths. And then beyond the visible light spectrum, we start talking about things like ultraviolet, X-rays, and cosmic rays. These have shorter and shorter wavelengths. And because their wavelengths are smaller, that radiation is more likely to be absorbed by the molecules of your body, and that can lead to basically molecular damage. So like UV exposure, and UV is actually a component of sunlight, that itself can damage some of the molecules in your body. Otherwise, exposure to things like X-rays and other cosmic rays like gamma rays can also lead to molecular damage. So it's important to limit your exposure to the UV and X-ray spectrum. Now what happens with exposure to radiation injury is that some forms of radiation are ionizing. And if they're ionizing, what that means is it causes ions to form. It actually can cause molecules to break apart and ionize. What this allows for then is actually free radicals to be produced. And free radicals are basically molecules that have an unpaired electron. And what this molecule does is it goes through and starts making and breaking chemical bonds throughout the cell. I liken free radicals to a toddler with a sledgehammer, just going through a cell and just breaking things apart kind of indiscriminately and wildly, and that can lead to irreversible cell damage. Now, ionizing radiation also affects DNA, because if you start breaking apart DNA strands, then that can lead to formation of mutations that can lead to things like cancer or even cell death. Now, some of the symptoms and signs of acute radiation sickness are things like cerebral edema, esophagitis, myocarditis, pulmonary inflammation, kidney inflammation, bowel necrosis, dermatitis, bone marrow suppression, and sterility. So think about the tissues in your body that divide a lot, especially things like epithelial tissues. Those are going to be affected by radiation, more dramatically. So that's why we're going to see things like esophagitis, myocarditis, pulmonary inflammation, bowel inflammation, and kidney inflammation, because those are full of epithelial tissues. You're going to see sterility as well, because radiation exposure can affect sex cell division. And so if you get exposed to too much radiation, that can lead to sterility by damaging your sex cells. And the reason why you see bone marrow suppression or depression is that the cells in your red bone marrow that are dividing rapidly, those can be particularly damaged by radiation exposure, which can lead to things like leukopenia, anemia, and thrombocytopenia. Leukopenia just basically means low levels of white blood cells. Anemia just basically means low levels of red blood cells. And thrombocytopenia means low levels of platelets. So basically, you're going to see low levels of all of these blood elements due to bone marrow suppression. Now, in terms of aging of cells, what happens is that typically what we see is that there's this progressive decline in the ability of your cells to proliferate and thrive. Basically, as your cells start to age, certain processes break down. Your cells don't divide as rapidly, and they're not able to accommodate for different changes in environmental factors. And you're going to see a lot of accumulation of things like cellular and molecular debris and damage. Now, what happens is that typically cellular aging is relates to DNA damage, because your cells can only divide a certain number of times before certain genes start breaking down, so that over the course of a lifetime, after the cells of your body have divided, numerous number of times, genes start being sort of cut shorter, and the cell stops being able to function properly. So what we see here is the length of a given chromosome, and we see that the ends of our chromosomes have basically just a length of DNA which we call telomeres. Telomeres are sort of non-coding segments of the chromosome that they're not really genes that encode for anything. They're just extra DNA, because every time you copy a chromosome, it's copied, but a little bit's left behind. So you can't copy the full length of that chromosome. There's going to be a small segment of DNA that can't be copied. And that's just due to the nature of the copying mechanisms of our DNA. So you see that once this chromosome's copied, it's a little shorter. When it's copied again, it's even shorter. So every time the cell divides, the chromosomes become shorter. Now, eventually what happens is the telomeres become shorter and shorter. And once these telomeres start to totally disappear, what that means is the next time the cell divides, you're now dipping into the parts of the chromosome that have your genes. So that now you start to see gene damage as a consequence of too many cell divisions. And this can lead to cell death. And this is one of the mechanisms our body uses to avoid cancer. You think about it, as your cell divides a certain number of times, there's an increased potential to accumulate mutations. And this is sort of like a built-in mechanism to prevent cancer. However, it also leads to a certain lifespan, because your cells can only divide a certain number of times. So there's people who are trying to study how to increase the length of these telomeres. That way your cells could have like an indefinite lifespan, and that could be like the fountain of youth. You could have people living for hundreds or thousands of years if you can keep these telomeres long. However, those individuals would run the risk of higher rates of cancer due to the accumulation of things like mutations.